Do you know the genetics behind pigment formation in plants? Welcome to your own channel ADP for Learning. I am your own learning partner Aashish. Let's begin our journey regarding the genetics of flower color. Flower color is the result of pigment molecules accumulating in cells, but it's not as simple as just making pigment. The location, type of pigment, and amount produced are all very important. These aspects are genetically controlled. Two main groups of genes control flower color. One group includes genes that code for the protein machinery required to make pigment molecules. The other group includes genes that code for regulatory proteins. It's the regulatory proteins that control the location, type, and amount of pigment-producing machinery made. In flowers, anthocyanins and carotenoids are two of the major pigment types. They are built by a series of chemical reactions within cells. Some flower color genes code for the protein machinery directly involved in the reactions. These proteins are called enzymes. An enzyme's job is to catalyze a biochemical reaction. They facilitate the attachment, removal, or rearrangement of a molecule's chemical groups. To build a pigment molecule, different enzymes make specific changes, one after the other, until the final product is formed. Enzymes working together in this way form the core of a biochemical pathway. A single biochemical pathway can require dozens or even hundreds of enzymes. In flowers, different groups of enzymes work together to make anthocyanin or carotenoid pigments. A specific set of genes code for the enzymes of the anthocyanin biosynthetic pathway. Another set code for the enzymes of the carotenoid biosynthetic pathway. Anthocyanins and carotenoids are made by different protein machinery and have different structures. You can check my previous video, The Magic of Orange, exploring the biochemistry of marigold flowers, to learn how the structure of pigment molecules determines the colors we see. The link to the video is available in the description. Now, coming back to our main topic of the genetics of flower color. A second group of flower color genes code for proteins called transcription factors. Transcription factors are regulatory proteins. Each cell type has a unique set of transcription factors that control gene activity. In different types of flower cells, transcription factors turn genes in the anthocyanin and carotenoid pathways up or down. They also control the timing of gene activity. That's why even though all flower cells have the same genetic content, they aren't all the same color. Transcription factors work through genetic switches. There are one or more switches controlling every gene. The switches aren't genes, but they're small stretches of DNA that affect gene activity. Transcription factors bind to a gene's switch, turning gene activity up or down. So, after knowing this, a question may come to your mind: which gene makes a monkey flower appear pink or yellow in color? To answer this, let's explore how the monkey flower, whose scientific name is a bit tricky, that is Mamulus lewisii, gets its color. It acquired the name monkey flower due to the shape and appearance of its blossoms, which some people thought resembled the face of a monkey. The flowers have a tubular shape with two lips, and they often display bright and vibrant colors such as yellow, orange, red, or pink. The overall structure of the flower, particularly the lower lip, has been compared to the face of a monkey, hence the common name, monkey flower, for Mamulus lewisii. Monkey flower flowers have pink petals from anthocyanins. They also have two yellow nectar gids, colored by carotenoids. In a pink petal cell, transcription factors activate genes of the anthocyanin biosynthetic pathway. Many genes in the pathway have a similar switch, so they respond to the same transcription factors. Activated genes produce the protein enzymes they code for. The enzymes do their job of making anthocyanin pigment, and the petals are pink. So far, researchers know 3 transcription factors that activate anthocyanin biosynthetic genes in monkey flowers. They are called MIWD40A, MLANBHLH1, and pelin. All 3 work together as a group to activate genes. A similar story explains why monkey flowers' nectar guides are yellow. A transcription factor protein called RCP1 is in nectar guide cells, but not the other cells of the petal. RCP1 switches on genes whose protein products make carotenoid pigments. The nectar guides are yellow. As shown in figure, in most petal cells of this monkey flower, transcription factors activate genes in the anthocyanin biosynthetic pathway. The cells make proteins that build pink pigments. In nectar guide cells, transcription factors activate genes in the carotenoid biosynthetic pathway. Proteins are made that build yellow pigments. In reality, it's not just on or off. The monkey flower example describes how transcription factors activate genes, but they can also turn gene activity down. In the end, it is rarely as simple as turning the genes in a pathway on or off. It is usually a balance. A single cell type can also make more than one type of pigment, and levels can vary greatly. This complexity is part of what allows for the huge variety of flower colors that exist. The petals of monkey flowers usually have a combination of transcription factor proteins. They are light pink because some transcription factors turn gene activity up, while others are turning it down at the same time. When one type of transcription factor is removed, the balance of gene activation shifts. Flower color changes. There are other examples like coloring fruits and vegetables where it's not just on or off of genes. As shown in the figure, transcription factors help control coloring in many plants, including fruits and vegetables. So, the question may come to your mind. What activates the transcription factors? The monkey flower example describes how transcription factors activate genes, but they can also turn gene activity down. In the end, it is rarely as simple as turning the genes in a pathway on or off. It is usually a balance. A single cell type can also make more than one type of pigment, and levels can vary greatly. This complexity is part of what allows for the huge variety of flower colors that exist. Transcription factors are also sensitive to signals from the environment. The signals can affect the amount of a transcription factor protein that is created, or ensure the protein acts at the right time and place after it is created. Cues from an organism's development, a cell's communication with its neighbors, and environmental signals like light, stress, and temperature can all affect transcription factor activity. As shown in the figure, the color of blood oranges is due to a mix of carotenoid and anthocyanin pigments. They contain an anthocyanin-activating transcription factor that is more active at low temperatures. This means anthocyanin accumulation and blood orange's color is different depending on temperature. Now, coming back to solving the old puzzle of monkey flower. Because we understand so much about the genetics of color in monkey flowers, we can use the knowledge to guide research into other questions. For example, researchers use monkey flowers to study overdominance. Overdominance is when the hybrid offspring of two parents have traits that are more extreme compared to either parent. In plants, an inbred line consists of individuals that are genetically very similar. They are established by either generation of strict crosses within the line, or self-pollination. Crossing two inbred lines gives hybrid offspring. As shown in the figure, this hybrid monkey flower is darker than either of its parents. It occurs in many different plants and animals, but it is very well known in crops. Hybrid crop plants often have higher yields than their purebred parents. Farmers have used hybrid crops to improve yield for over a century, without fully understanding why it works. Knowing more might help us boost its effects. Now, let's understand the concept of overdominance in monkey flowers. In the monkey flower, overdominance affects the intensity of flower color. If two specific inbred lines, one with light pink petals and one with very pale petals, are crossed, the hybrid offspring are dark pink. Researchers discovered the dark pink monkey flower is a case of single gene overdominance. It is dark pink because it has two forms that is alleles of a single gene. It gets one from each parent. The gene responsible for the overdominance is flavone synthase or FNS. It codes for the protein FNS. FNS interferes with an enzyme in the anthocyanin biosynthesis pathway. Both FNS and the enzyme use the same building material but make different end products. FNS takes building material away from the anthocyanin pathway and uses it to make flavones. Flavones are also important for monkey flower color. Once made, they bind and stabilize anthocyanins, preventing their breakdown. You can see the strength of hybrid in crop plants. An example of this you have seen in hybrid tomatoes that often produce more fruit. In crops, hybrid vigor, also known as heterosis, describes hybrid offspring that do better than their parents. For a long time, the molecular basis was unknown. Now we know at least a few cases are because of single gene overdominance. As quoted earlier, the example is in tomato yield. The gene single flower truss or SFT leads to the production of florigen protein, a hormone that tells plants that it is time to make flowers. Tomato plants are very sensitive to the amount of florigen produced. Too little, and they don't produce many flowers. Too much, and flower production slows down or ends. However, with one working and one non-working version of the SFT gene, Florigen protein is produced at a level that allows plants to make more flowers. With more flowers, there are more tomatoes. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.